For more than 2 decades, biology textbooks have told a very specific story about how our cells divide. Imagine a microscopic ballet where tiny structures called chromosomes, which hold all our genetic information, must line up perfectly in the middle of a cell before it splits in two. Scientists believed a protein named CENP-E was one of the main dancers, a powerful motor that physically pulled these chromosomes into place. It was seen as a tiny locomotive, chugging along cellular tracks, and dragging its precious genetic cargo to the center stage. This idea made a lot of sense, as it explained the powerful forces needed to organize a cell's most vital components with such precision. This long-held belief painted a picture of brute force, where CENP-E acted like a molecular tugboat, ensuring that each chromosome arrived at its destination. The process, known as mitosis, is fundamental to life, growth, and healing. Every time you heal from a cut, or grow a little taller, it's because your cells are dividing correctly. The idea that CENP-E was a simple puller was a cornerstone of our understanding. It was a neat, tidy explanation for a complex process. But as we all know, science is a journey of constant discovery, and sometimes the most established ideas are the ones that are ready to be turned completely upside down. Now, thanks to a team of brilliant researchers in Zagreb, Croatia, we have to get out our pens and rewrite that part of the textbook. A groundbreaking study from the Ruđer Bošković Institute has revealed that CENP-E isn't the muscle of the operation after all. It's more like the brain. The scientists, led by Dr. Kruno Vukušić and Professor Iva Tolić, use powerful microscopes and advanced techniques to watch this cellular ballet in real time. What they saw was astonishing. Instead of pulling chromosomes across the cell, CENP-E's primary job is to make sure the chromosomes get a good, solid grip on the cellular tracks in the first place. This changes everything. This isn't just a minor tweak to the story, it's a complete plot twist. The discovery, published just last month on October 21, 2025, in the prestigious journal Nature Communications, forces us to rethink the very mechanics of how life duplicates itself. It shows that the initial connection, the handshake between the chromosome and the cell's internal skeleton, is the most critical step. CENP-E is the master of this first introduction, ensuring the connection is stable and secure before the heavy pulling even begins. Without this crucial first step, the whole process of cell division would grind to a halt, with chromosomes lost and unable to find their way. To understand this new role, let's think of the cell as a huge, bustling city. The chromosomes are like very important delivery trucks, each carrying an irreplaceable package of genetic data. These trucks need to travel from warehouses near the city's edge to a central distribution hub before the city can expand. The roads they travel on are called microtubules, long protein filaments that form the cell's transportation network. For a long time, we thought CENP-E was the engine of the truck, powering it down the road. But this new research shows it's something much more subtle, and frankly, much more clever. CENP-E is the traffic controller. In this new model, CENP-E's job is to stand at the on-ramp where the chromosome truck first tries to merge onto the microtubule highway. It's a chaotic intersection. Other proteins, called aurora kinases, act like flashing red lights, signaling, "Stop! Don't attach here!" This prevents the truck from connecting to the wrong road, or at the wrong time. CENP-E's job is to step in at the perfect moment and say, "Okay, this is the right spot. Go ahead and connect." It essentially overrides the stop signal, allowing the chromosome to form a stable, end-on attachment to the microtubule track, like a train car coupling perfectly to the rails. Without CENP-E acting as this skilled traffic manager, the chromosome trucks would get stuck. They would drive up to the highway on ramp, see the flashing red lights of the aurora kinases, and just sit there, unable to merge. As the Croatian scientists observed, cells without functioning CENP-E had chromosomes that lingered at the edges of the cell, failing to line up at the center. They couldn't make that first, crucial connection to the transportation network. So, CENP-E isn't the engine pulling the train. It's the sophisticated coupling mechanism that ensures the train gets on the track properly to begin its journey. This shift from a puller to a connector is a beautiful example of the elegance found in nature. It's not about brute force, but about smart regulation. The cell uses a system of checks and balances with stop signals, aurora kinases, and go signals, CENP-E, working in harmony. This ensures that each chromosome, with its precious genetic cargo, is handled with extreme care. The process has to be perfect every single time, in trillions of cells throughout our lives. This intricate system of signals and stabilizers is what makes that incredible feat of precision possible, all orchestrated by proteins like our newly understood friend CENP-E. So, why does this change in our understanding of a single protein matter so much? Because when cell division goes wrong, the consequences can be devastating for our health. Think about it. If the chromosomes, the blueprints for our entire body, are not divided equally between the two new cells, it leads to chaos. One new cell might get too many chromosomes, while the other gets too few. This condition, called aneuploidy, is a hallmark of many serious diseases. It's like trying to build two identical houses, but you accidentally give all the window frames to one construction crew and all the doors to the other. Neither house will turn out right. This is especially critical when we talk about cancer. Cancer is essentially cell division running out of control, and it is often riddled with errors. Cancer cells frequently have the wrong number of chromosomes, which helps them grow uncontrollably and resist treatment. Now that we know CENP-E is a key regulator of chromosome attachment, we can see it as a potential weak spot in the cancer cell's machinery. If we can develop drugs that specifically target the function of CENP-E, we might be able to selectively disrupt cell division in tumors, causing those cancerous cells to self-destruct while leaving healthy cells unharmed. It's like finding the master switch for the faulty traffic control system in the city of a tumor. Beyond cancer, errors in chromosome segregation are also a leading cause of genetic disorders and developmental problems. Conditions like Down syndrome, for example, are caused by an extra copy of a chromosome. Many miscarriages are the result of embryos having an incorrect chromosome count, making them unable to develop properly. By understanding the precise molecular steps that ensure chromosomes are segregated correctly, we open the door to new diagnostic tools. We might one day be able to screen for subtle weaknesses in the cell division machinery and predict the risk of these conditions, offering better guidance and potential interventions for families. This discovery about CENP-E, therefore, isn't just an interesting fact for a biology textbook, it's a vital clue in our ongoing mission to understand and fight human disease. It provides a new target for drug developers and a new window for researchers to peer through. By mapping this critical vulnerability in the process of life's continuation, the scientists at the Ruger Bošković Institute have given the medical community a new direction to explore. This fundamental knowledge is the bedrock upon which future life-saving therapies will be built, all thanks to a fresh look at a tiny protein with a very big job. This kind of breakthrough doesn't just happen in a flash of inspiration, it is the result of a modern scientific process that looks a lot like a symphony, with many different players and instruments all working together in harmony. The discovery about CENP-E was made possible by incredible teamwork. The Zagreb-based team, led by Dr. Vukušić and Professor Tolić, collaborated, shared ideas, and challenged each other's assumptions to get to the truth. This collaborative spirit is the engine of modern science, pushing the boundaries of what we know by combining diverse skills and perspectives to solve a single, complex puzzle. These scientists also had powerful tools at their disposal. To watch something as small as a protein inside a living cell, you need incredibly advanced microscopes that can see things on a molecular scale. Furthermore, analyzing the vast amounts of data generated by these experiments requires immense computing power. The researchers relied on high-performance computing infrastructure to model the cellular mechanics and confirm their observations. This combination of brilliant human minds and cutting-edge technology is what allows us to see the inner workings of our cells with a clarity that was unimaginable just a generation ago. Of course, none of this would be possible without funding. Big, ambitious science requires significant investment. This research was supported by prestigious grants from organizations like the European Research Council and the Croatian Science Foundation. These grants are awarded to the most promising and daring ideas, giving researchers the freedom and resources to pursue fundamental questions without the pressure of an immediate commercial outcome. It's an investment in knowledge itself, based on the understanding that today's basic research is the foundation for tomorrow's life-changing applications. It's a perfect example of how public and private investment in science pays off in the long run. In the end, this story shows us that even a small change in our scientific understanding can have massive ripple effects. Redefining the job of one protein might seem minor, but it fundamentally alters our view of a process essential to all life. It opens up new avenues for treating diseases that affect millions of people worldwide. It reminds us that science is never settled. It's a dynamic, evolving quest for knowledge, and every discovery, no matter how small it seems, adds another piece to the grand puzzle of who we are, how we work, and how we can live healthier lives. And that, for all of us, is a very big deal.